Over the next few minutes, I'm going to talk about cooling for hypoxic ischemic encephalopathy, and I'm going to give the Indian perspective. The burden of the problem in India, unfortunately, the last national data that we have is from the NNPD database in about 2003, where it said that 14 of 1,000 live births, babies developed HIE, and this would translate to about 364,000 babies with HIE in India per year, and that's a massive problem. And asphyxial deaths accounted for nearly 30% of neonatal deaths in India, and I think this still holds good in most parts of India. As we know, what are the problems with asphyxia? All of us do know that there is a high rate of cerebral palsy. In the time before cooling started, analysis of 8 studies showed a high rate of cerebral palsy of very variable rate of cerebral palsy between 23 to 82%, but it doesn't end there. These babies who survive do have decreased IQ at 2 to 6 years. Their school performance is affected. They can go on to have autism spectrum disorders, aggression, anxiety, and the Finnish birth cohort study also linked asphyxia with an increased risk of developing adult onset schizophrenia. Unfortunately, the only intervention after birth which can modify at least the long or the medium short-term outcome is therapeutic hypothermia. And this was extensively studied in animal models before clinical application in humans. And then the animal experiments were done in the 1990s, and since then we've had large RCTs in Europe, US, Canada, Australia, New Zealand, and the UK between 2000 and 2010. This is the Cochrane review in 2013, which decisively proves that therapeutic hypothermia causes a reduction in the risk of death or major disability, and also there is less neurodisability in survivors at the age of 18 months. Now we have the childhood outcomes from both the TOBY trial as well as the NICHD trial, and we have similar outcomes. Survival in the TOBY trials follow up at 6 to 7 years. There is a definitely significantly increased outcome of survival with a better IQ at 6 to 7 years, and there was also a 15% reduction in cerebral palsy and moderate to severe disability. Similarly, in the NICHD data, the childhood outcomes showed that there was significantly less death or severe disability at between 5 to 8 years. I'll just shortly dwell upon the journey in therapeutic hypothermia in India. In the mid-noughties, so about 2005 or so, these were the equipment which were available for cooling. The Olympic Cool Cap, the Ticotherm, the Blanket Roll, the Criticool, all of them, the price varied between 16 lakhs to 25 lakhs. That is about 1.6 million to 2.5 million rupees. Okay, and all of them were expensive, so they were cost prohibitive to most units in India. They needed continuous electricity, which again is a problem in most parts of India, and there was a need for ongoing disposables. My colleague Niranjan Thomas he was a participant in the ICE trial in Canada at that time, and when he came back, he thought, why don't we start trying to cool with cool gel packs? Okay, so we started a feasibility trial in 2007 using cloth-covered vaccine ice gel packs. This started in 2007, and we recruited 20 babies. We finished the study in 2009, and this was published in the Indian Pediatrics in 2010, where we proved that it was feasible to safely cool babies and maintain this cooling for the duration that it should be maintained. But the problem with the vaccine cool gel packs, we found that we needed to frequently change these cool gel packs. It was very nursing intensive. We needed a lot of nursing input, needed constant monitoring. There was more shivering in these babies as opposed to babies who were cooled in the larger RCTs with servo-controlled methods, and there were larger swings of temperature, which again is not very ideal. Following this, the team from JIPMER actually used the same vaccine gel packs and the same protocol, and they did the first RCT in India, and they published that in 2012, where 65 babies were there in the hypothermia and the control arms, and the primary outcome was mortality and 6 months development. And both of them, they proved that there was an improvement in primary outcome in the cooled babies as opposed to the non-cooled babies. Since then, this group from JIPMER has published 4 different RCTs. As I said, vaccine cool gel packs, we found that there were problems when using vaccine cool gel packs. So we were working on some other method of cooling. And between 2011 and 2012, we actually tried devising a servo-controlled device using a Peltier effect. This in the laboratory seemed to work, but we did not carry it on till human trials. There were some concerns about safety of this method. Simultaneously, my colleague, again, Niranjan, worked on phase change material between 2011 and 2013. Phase change materials are either organic or inorganic materials which have a specified melting point. Okay, and they either emit heat or they absorb heat from any surface in contact with them, and finally they act, and therefore they sort of neutralize the temperature, and therefore they will act as natural servo-controlled devices. And this has been used in industry in many instances. Iwata in 2009 actually published a study using PCM mattress, and he proved that it could produce effective cooling in a piglet model. Sudhan Thayil, again, he started and completed a phase 2 trial, RCT in Calicut. Some of you might be aware of that, where they cooled 17 babies using the PCM mattress. Unfortunately, in this study, he found that the mortality was greater in the babies who were cooled, and they also felt that the PCM was effective only when the ambient temperature was below 28 degrees Celsius. We worked on a PCM mattress, and over 2 years, there was some trial and error, and finally we came out with this mattress using 2 PCM gels of different melting points, and this was the CMC PCM mattress, which finally came to fruition in 2013. Right, and these were the babies who were cooled by the PCM mattress, and this was published in Neonatology in 2015. 41 babies were cooled using the CMC PCM mattress, and we found that the mean temperature, we could maintain it quite well at the desired temperature. The rate of rewarming was also good, and the target temperature, we could maintain it 96.2% of times, with babies falling below the target range in 2.4% of times and greater than the target range 1.4%, which is fairly, comparable to other studies. Since then, the company which actually supplied the PCM, that was Plus Polymers, they took over the technology, and they are now marketing this technology as the Mira Cradle, and it's now being used extensively in India. It's got CE certification, so it's starting to be used in Europe and also in South Africa. And this is the Mira Cradle, a baby in Mira Cradle. Currently in India, again, there is a trial which is called Hypothermia for Encephalopathy in Middle and Low-Income Countries, Helix trial, which is using a servo-controlled device, the Ticotherm Helix, and this is currently underway. The protocol says that they want to randomize 408 newborn in India, Bangladesh, and Sri Lanka, with the primary outcomes being death or moderate or severe disability at 18 months. And now manual and servo-controlled devices, Ticotherm, Blanketrol, Criticool, are freely available in India. So cooling in India, like I said, the units which started, CMCV started in 2007, JIPMER started cooling in 2009, Ramachandra Medical College in 2010, and a few other hospitals around that time. More than 600 babies have been cooled, either with the vaccine gel pack or with the PCM mattress. And the mortality, the nursery mortality during this time has been in these studies between 8 to 25%. Having said all this, there was a article in the Journal of Pediatrics in 2012 giving the recommendations of an NICHD workshop. And they expressed some concerns about hypothermia in low resource settings. They felt that brain injury in low resource settings may occur at long intervals before birth. This might be because the mothers got admitted late when they had a morbidity, delivery was delayed beyond the winter period, and even if the babies were asphyxiated, transport systems were not adequate to transport these babies on time to receive hypothermia, and therefore the window for hypothermia may be missed. They felt that there was a high incidence of early onset sepsis and infection in developing countries or low resource settings, and cooling in infection may be deleterious. They felt that, definitely cooling would be unsafe when the babies had coexistent meconium aspiration or PPHN, because many centers in these countries would lack facilities to tackle multi-organ dysfunction, and cooling equipment may be expensive. And therefore, the recommendation was careful RCTs were needed in low resource settings to prove safety and efficacy of cooling before cooling was started as standard of care. And they wanted that access to quality obstetric and newborn care should be mandatory before cooling was started. We did agree with a few of these recommendations, but we actually wrote a counter in the Journal of Pediatrics in 2012. For one thing, we felt that we were not sure in technology which had been clearly proven useful, whether it was ethical to actually withhold this when we had proved the feasibility and safety before this publication came out. We felt that a registry or ongoing thing of observations would probably help. We did agree with the fact that obstetric care and newborn care should be improved, and I'll come to that later on. And therefore, I'm going to answer a few questions. One, is it safe to cool in the Indian context? Is it feasible to start and maintain cooling within the therapeutic range of 33 to 34 degrees Celsius? And could we start within the window period of the first 6 hours? Do we need to adapt or change protocols during cooling in India? Cooling in a resource-poor setting, should we or can we use alternate methods? And whatever I'm going to speak further on is using alternate methods. Should it be the standard of care in India, and should efficacy be proved in India separately? Can we use cooling in a secondary level setting, and alternate strategies for those who come too late to cool? Right? Talking about feasibility, these were the temperatures which were seen in the NICHD trial, the TOBY trial, the ICE trial, and the trial by Horn. And this was the temperature which was seen during when we cooled the 41 babies initially using the PCM. And you can see that we could maintain temperature within the target range. Okay? And time to reach the desired temperature when we use the cool gel pack, it did take longer, 90 minutes with an interquartile range of 25 to 150 minutes. Whereas when we started using the PCM, we could cool within 45 minutes with an interquartile range of 10 to 100 minutes. And temperature outside the target range with PCM was only 3.8% as compared to 9.8% of times when we used the vaccine cool gel pack. Safety, is it safe to cool with alternate methods? I'm just talking about few of the side effects seen after cooling: thrombocytopenia, altered coagulation parameters, PPHN, arrhythmia, sepsis, hypotension in the NICHD TOBY trial, the vaccine gel pack used by JIPMER, and the PCM. You can notice the one thing that I highlighted is that sepsis is not greatly different in the Indian trials as compared to the NICHD and the TOBY trial. The only thing we found was, for some reason, we had a higher degree of altered coagulation parameters. One of the reasons could be that when we initially started cooling, we had to discontinue cooling, and we had 2 deaths because of uncontrollable bleeding. Since then, we have started actually doing coagulation parameters before we start cooling, and we preemptively give the baby's plasma, and therefore it is possible that because we did coagulation parameters before cooling and then continue to estimate coagulation parameters, that our incidence is higher, right? There is a meta-analysis which was published in 2015 of 3 trials involving 467 infants using low-tech methods, and this proved that death before discharge was definitely reduced, reduction in neurological morbidity at discharge, and there was a decrease in mortality or severe morbidity at 24 months. So cooling using low-tech methods is effective. Right? CMCH data, we have been cooling since 2007 in the feasibility trial. We cooled 20 babies, and then since then we have a cooling registry. We have cooled about 117 babies in the next 8 years or so. Adverse outcome before discharge, there were 23 babies who had an adverse outcome. 86 babies have been followed up to 18 to 24 months. At 18 to 24 months, 9 of 86 had an abnormal neurological outcome, and death and disability together, there were 31 babies who had a thing. I've not given the denominator because this includes data from 2 trials, the details of which have not been published, and we are probably going to get the results next year. Of 60 babies not cooled during this time, 16 of 60 had abnormal neurology at 18 to 24 months. Right? The next question is, can we cool in secondary settings? This is a question which has been frequently asked when we actually talk about cooling. A meta-analysis by Prakash Shah showed that there was a significant increase in arrhythmia, thrombocytopenia, pulmonary hypertension, coagulopathy, trend towards increased coagulopathy in babies who were cooled. In our experience, 25% of asphyxiated babies required respiratory support. Of babies who died, 15% had coagulopathy, 10% had PPHN, and 15% had multi-organ dysfunction. And more than 50%, like I said, had abnormal coagulation parameters before or during cooling. And these can only be managed in a tertiary center. The other question is people try to find shortcuts. The one thing is if they are not able to continuously monitor rectal temperatures, could they use skin temperature as a surrogate for rectal temperature? So in the feasibility trial, when we had taken multiple readings of temperature, we looked at the correlation between rectal temperature and skin temperature. And you can see that the correlation seems to be absolutely beautiful. But when we looked at the agreement between rectal and skin temperature, we found that there was a difference of plus 6.63 degrees Celsius and minus 7.75 degrees Celsius. And this is absolutely not acceptable when you're trying to keep babies between a narrow therapeutic range of 33 to 34 degrees Celsius. And therefore, skin temperature cannot be an alternative to rectal temperature. Rectal temperature has to be, or esophageal or rectal temperature have to be the modes by which babies are monitored during cooling. Dr. Diorari had mentioned that you need structure and process, and then only you'll get a good outcome. Therefore, the same thing I'll reiterate. It's just not enough to cool the babies and expect a good outcome. You need to have good obstetric care. When we started cooling, maybe in the early 2000s, our asphyxia rates were about 3.4 per thousand. Simultaneously, from the time that we started cooling, our obstetric department has taken a series of steps. Over the last 2 years, our asphyxia rates are less than 1 per thousand. And the same thing with Ramachandra. Babies who have been exposed to better antenatal care are probably, they have less morbidity when we start cooling, and they do have less morbidity after cooling, and their long-term outcome also will be better. So we need multi-parameter and other monitors, we need ventilators, we need blood products, and we need intensive nursing. And therefore, cooling in secondary settings where these cannot be provided, I don't think should be done. Okay. Do we need to change protocols? Different criteria for outborn babies. As you know, in India, outborn babies will come with no Apgar scores and no blood gases. And therefore, we need to have different inclusion criteria for outborn babies. We found while we started cooling that babies who were cooled tend to have higher levels of phenobarbital. And so slowly over time, we have changed our policy and we give only one bolus of 20 milligrams per kg of phenobarbital. Anything above that, babies' phenobarb levels go to toxic levels, so no maintenance phenobarb, only 20 milligram per kg of phenobarb. And we do coagulation profile prior to cooling, and after we started doing that, we have not had uncontrollable bleeding in any of our babies. And therefore, whom to cool any difference, you'll notice in the outborn babies, we have given, if present, we give Apgar score. The inclusion criteria is almost similar to the TOBY trial for inborn babies. The outborn babies, second criteria is Apgar scores if available at 5 minutes of less than 5. But then we have added another thing of needing resuscitation or bagging for 10 minutes, or history of not having cried at birth, not breathing normally at 5 minutes at birth, and given assistance for breathing or limp or flaccid since birth. The remaining concerns, like I said, there needs to be improved obstetric and perinatal care before you start a cooling program. Outborn babies, again, is there a problem with cooling outborn babies? One of the Norwegian pediatric residents did an audit in our unit, and over 5 months when she stayed with us, she found that of 53 babies who had asphyxia, 45 of them arrived beyond the therapeutic window. So the question is, do babies who come after 6 hours, can they be cooled? Can we give late therapeutic hypothermia? Can we start therapeutic hypothermia in transport, or should we try for early referral? It is good for units which cool to have a fixed protocol. You should know whom to cool, when to cool, how to cool, when do we withdraw cooling, and what should be the protocol for follow-up. It will be great if we have a registry. Like I said, the 4 units which I had presented before, all of us follow the same protocol for cooling, but we have been striving for a registry, at least in Tamil Nadu and South India, and this is a dream. And we need to have better follow-up. So late hypothermia, I'll just briefly tell you the thing. There is a study on late hypothermia between 6 to 24 hours. Dr. Sita Shankaran is the prime investigator. 168 infants in 16 NICHD sites are being monitored. The study has been completed, but the results are not yet out. I got some data from the clinical trials website where it says death or moderate disability doesn't seem to be very different, and therefore I'm not sure that late hypothermia would help. This is the protocol in our unit: AEG during cooling, MRI followed by day 5 and day 7 neurological examination, and then general movement assessment we used to do as part of a research project at 1 year P-body test, and Bailey at 18 to 24 months. We have just started 3 months, and preschool and school age evaluation of these babies. This is the Vellore team, and the person there, the second to the left is Niranjan Thomas, who's done most of the work on cooling in my institution. These are some of our babies. And therefore, is it safe cooling in the Indian environment? Yes, it's in the right environment, it's safe. Is it feasible to start and maintain cooling in inborn? Definitely yes. Still an issue with outborn to start cooling in the right time. Do we need to change protocols? We have changed inclusion criteria for outborn. Cooling in a resource-poor setting, can we use alternate methods? I think so, yes, but monitoring will not change. Should it be the standard of care? A registry should help in saying, giving us further data on efficacy of cooling. The Helix outcomes, when they come, can probably give us also more data. And can we cool in secondary settings? No, at the moment. I'll stop with that. Thank you.